studies utilizing ancient DNA have just been getting cooler and cooler. I'm sure just a couple decades ago, nobody would have thought that we'd have DNA from ancient Egyptians, Pompeian victims, or Vikings. The genetic history and prehistory of Rome also belongs on this list. This paper, published in 2019 in the journal Science, analyzed the genomes of individuals buried in Rome and the region around it over a 12,000-year time span. It covers the Mesolithic all the way to the modern day, and gives us a rare insight into how the population centered around what would become the capital of one of antiquity's greatest empires shifted over time. I'm Madam Archaeologist, your go-to informant on everything archaeology, and in this episode, I'll discuss an ancient DNA study on individuals who were buried at sites and cemeteries in Rome and in the region around it from about 10,000 BC onwards. I want to note before diving into the findings that I am mainly discussing the average here. Of course, there will be genetic differences from person to person, but what I am looking at here is the general trend. I also want to make it clear that I'm not discussing Italian genetics, I'm just focusing on the genetic history and prehistory of the area around Rome. And I'm sure, if you haven't read the paper, that you'll encounter some cool surprises. First up, we've got the Mesolithic period. The Mesolithic refers to the last period of hunting and gathering in Europe. It started around 10,000 BC and ended with the introduction of agriculture. Agriculture reached different areas of Europe at different times, and so the Mesolithic spans a different length of time depending on the region, but in Italy it ran up until about 6,000 BC. In this study, we've got 3 Mesolithic hunter-gatherers that date to about 12 to 9,000 years ago, and they come from a cave site known as Grotta Continenza. They cluster together here on the PCA with what we call Western hunter-gatherers, or WHGs. We use the term WHG to refer to Mesolithic hunter-gatherers from southern, western, and central Europe. PCA, which you should be familiar with by now if you've watched my other videos, stands for Principal Component Analysis, and it is a very useful tool in ancient DNA studies because it gives us a way of visually seeing which population or populations, both ancient and modern, a sample is genetically closest to. In this more specific PCA plot, the Mesolithic hunter-gatherers cluster closest to WHGs from Sicily. Now, PCA is one of a few tools employed in ancient DNA studies. There is also admixture analysis, which gives us an idea of which populations a sample likely got DNA from, but admixture is not 100%, so a good archaeogeneticist will also run some statistical tests. One particular test that is very useful is called QPAtom, which, in simple terms, determines admixture events and sees which populations contributed their DNA to your sample. On admixture analysis, the Mesolithic hunter-gatherers came back as 100% WHG, but when tested with QPAtom, earlier hunter-gatherers from Villa Bruna in northern Italy and WHGs from Luxembourg, France, and Germany did not provide a good fit for the Mesolithic samples from Grotta Continenza, suggesting that there was some genetic differentiation between WHGs from central Italy and those in other locations. However, WHGs from the sample here, another previously published hunter-gatherer from the same cave, and one from Grotta d'Orient in Sicily form a clade, suggesting strong genetic similarities between hunter-gatherer groups from central Italy and those from southern Italy. However, since the latter samples had very low coverage, meaning that a good portion of their genomes couldn't be tested, we have to consider the possibility that there may have been some genetic differentiation between these hunter-gatherers that hasn't been picked up on yet. Regardless, a major genetic change took place during the Neolithic period with the transition to farming. Near Eastern crops and animals were introduced to Europe, and DNA studies from different parts of the continent confirm that this was associated with the actual migration of Neolithic Near Eastern farmers. Around Rome, the Near Eastern farmer DNA nearly overtook that of the local hunter-gatherers. Now, we call these Neolithic farmers that settled in Europe early European farmers, or EEFs. Ancient DNA studies tell us that EEFs came specifically from Anatolia. In most EEFs, the Anatolian farmer DNA is really strong. However, in admixture analysis here, the EEFs from around Rome also carried a significant Iranian Neolithic farmer or Caucasus hunter-gatherer derived ancestral component. This makes these EEFs a bit different from other EEFs who have had their genomes tested. In Cupiatum, the EEFs from around Rome were modeled as deriving about 5% of their ancestry from WHGs and about 95% of their ancestry from Neolithic farmers from central Anatolia or northern Greece, who themselves carried an additional Iranian Neolithic farmer or Caucasus hunter-gatherer derived ancestral component. And so that Iranian Neolithic farmer component was probably already carried by the EEFs who settled in central Italy. During the late Neolithic and the Copper Age, WHG ancestry increased a bit, but the EF DNA is still very strong. On average, the WHG component was found to be around 6 to 9%, so it's still small, but it looks like this extra WHG DNA came from admixture with farming groups carrying more WHG ancestry rather than direct admixture with hunter-gatherers themselves. Unfortunately, the Bronze Age is missing in this paper. This is unfortunate because this is when the second major genetic shift took place, when Western steppe herders from the Pontic-Caspian steppe moved westward into Europe. We call their ancestry steppe-derived. According to previous studies, Western steppe herders emerged out of a mix of Caucasus hunter-gatherers and Eastern hunter-gatherers, or EHGs. The best we have to assess the introduction of steppe ancestry in the region around Rome is the subsequent Iron Age. Admixture suggests that the steppe herders left a mark on the central Italian gene pool at that time, and that there was also an increase in Iranian Neolithic farmer ancestry. Cupiatum averaged the steppe component as an introduction of roughly 30 to 40%. The Iron Age was the period when we see the Etruscan and Latin city-states form, and we also see the emergence of the Roman Republic. The Iron Age exhibits some genetic diversity here. 8 out of the 11 individuals tested derived their ancestry from the preceding local Copper Age population and from steppe populations, but 2 of the remaining 3 individuals could be modeled as deriving their ancestry from the local preceding population and a Near Eastern population instead, probably related to either Bronze Age Armenian or Iron Age Anatolian populations. The 3rd individual from an Etruscan burial was, however, a surprising outlier, and I'll save that for near the end of the video. During the Imperial Period, that is when we see Rome transition from republic to empire, the main trend seen here is, I quote the paper, an ancestry shift toward the Eastern Mediterranean and with very few individuals of primarily Western European ancestry. The distribution of Imperial Romans in PCA largely overlaps with modern Mediterranean and Near Eastern populations, such as Greek, Maltese, Cypriot, and Syrian. This shift is accompanied by a further increase in the Neolithic Iranian component in admixture and is supported by F statistics. Compared to Iron Age individuals, the imperial population shares more alleles with early Bronze Age Jordanians and shows significant introgression signals in admixture F3 for this population, as well as for Bronze Age Lebanese and Iron Age Iranians. End quote. In other words, we're seeing an increase in ancient Eastern Mediterranean and ancient Iranian DNA. This is really interesting, especially that increase in ancient Iranian ancestry, because the Imperial Romans fought wars against the Sasanian Persians. If only they knew that they shared some DNA. Now, using Cupiatum, the researchers tested many two-way models for the Imperial Roman population. That is, they tested different combinations of ancestries using 2 populations at a time, one of those populations being the preceding local Iron Age population. They found that although some combinations were better fits than others, none of them were a good, solid fit with the data, suggesting that the ancestries of Imperial Romans from the bustling capital were complex. Diversity in the Imperial Roman population of Rome is expected. Rome at this time was the capital of an expansive empire which, at its height, stretched from Britain to Iraq and covered the entire Mediterranean. Still, the general trend we see in the capital at this time is this shift, genetically speaking, towards Eastern Mediterranean and Near Eastern populations, suggesting more influx of people from these regions into the capital than from the empire's western provinces. Our next period is late Antiquity. Major events within this time frame include the fall of Rome, the invasion and expansion of Germanic tribes, and the establishment of Germanic kingdoms throughout Europe, including Italy. Although I will note that the Roman Empire did continue in the east for about another 1,000 years after Rome fell, with its new capital at Constantinople. On the PCA plot, if you look at the green dots, we can see that there is diversity but also a general genetic shift away from the Mediterranean and Near Eastern populations and closer towards modern Western and Central European ones. On average, the late antique population of Rome could be modeled as deriving most of their ancestry from the preceding local population, and about 38 to 41% of their ancestry from that seen in either a late Roman Imperial Period individual from Bavaria or that seen in the present-day Basque population. Since it is not more specific than this, these combinations should be taken with a grain of salt. Even the paper cautions that, I quote, "The precise identity of the source populations and the admixture fractions should not be interpreted literally, given the simplified admixture model assumed and the lack of data for most contemporaneous ancient populations." End quote. However, at one of the late antique sites studied here, called Crypta Balbi, 5 of the 7 individuals that made up the sample could be modeled specifically as a mixture between the preceding imperial population of Rome and the ancestry seen in the Lombards buried in 2 cemeteries, one located in Hungary and the other in northern Italy. So, at least one of the ancient populations that contributed their DNA to the central Roman gene pool at that time was likely the Lombard population. This ties in with the historical record, as the Lombards established their own kingdom in Italy in the 6th century, and it lasted until 774. It's also possible that another reason the population generally shifted away from Eastern Mediterranean populations was because with the movement of the capital eastward to Constantinople, the influx of people from the Eastern Mediterranean region into Rome died down. During the Middle Ages and the early modern period, the genetic shift towards Western and Central Europeans continued, which you can see represented by yellow dots. The medieval Roman population derived their ancestry from the preceding late antique population and, on average, about 15 to 29% of their ancestry from another European population, which the researchers identified as potentially being either individuals from late antique or medieval Germany, Vikings from Sweden, Saxons from England, Lombards from Hungary, 6th century individuals from northeastern Spain, or an individual from Lithuania. Hopefully, one day, the exact source population or populations will be clarified. Quickly summarize the main trends I've discussed. Here is a graph showing ancestry proportions based on admixture analysis, which give us a rough idea of how the population centered in and around Rome changed over time. Blue represents Western hunter-gatherer derived ancestry, dark gray Iranian Neolithic farmer derived ancestry, orange steppe derived ancestry, and I'm not sure exactly what color this is showing as on your screen. On my phone it's green and on my computer it's light gray, but this is supposed to be Anatolian Neolithic farmer derived ancestry. We can clearly see the impact that Near Eastern farmers had during the Neolithic, where they almost completely overtook the local Western hunter-gatherer population. There is a small increase in WHG ancestry at the end of the period, but the Near Eastern farmer DNA is still strong. There is a gap with the Bronze Age, but we know that steppe ancestry was introduced, and we can see that component in Rome's subsequent Iron Age population. Some North African DNA is also seen, and we see an increase in Iranian Neolithic farmer DNA as well. We see a further increase in this Iranian Neolithic farmer component during the Imperial Period, probably with increasing connections to the Eastern Mediterranean region as Rome transformed from republic to empire, and then an increase in steppe-related ancestry during late Antiquity, which is likely related to Germanic tribes from northern and central Europe moving into the region. This trend continues into the medieval period, and in the very bottom row, we can see that all of the important events in Rome's history and prehistory all left their mark on the gene pool of modern-day Central Italians. This study is really cool because it focuses on one place over a very long period of time and allows us to see how the genetic ancestries of that place shifted over time. I also want to say again that this is the general pattern for the region. Of course, ancestries will vary from person to person, but what we are looking at here are the general trends. There were some genetic outliers identified in the study as well. An outlier is someone who doesn't cluster with the bulk of the population it was a part of. In other words, their ancestry doesn't fit the general trend. One particularly interesting outlier here is an Iron Age woman who was interred in the Etruscan necropolis of La Matonara near the town of Civitavecchia. Phonetically, she carried an unusually high component of African ancestry for the time and place. QP-Atom pumped out an ancestry composition model of approximately 31% of the preceding local Copper Age population, 16% steppe, and 53% Moroccan late Neolithic. Now, if we look at this chart from the study's supplementary materials, we can see 2 models the research considered as potential fits for this outlier. Both contain the local Copper Age population and steppe ancestry, but in one model the third population is Moroccan late Neolithic, and in the other it is ancestry seen in Mota from East Africa. However, when using QP-Atom, for a model to be considered a good fit, the p-value should be at least 0.05. Here the models are just under that. You can also see how the ancestral percentages change when you substitute Moroccan Late Neolithic for Mata. The African component goes down to 19%, and the local Copper Age component shoots up to about 70%. The researchers did note that they considered models with a p-value between 0.01 and 0.04 to be a marginally acceptable fit. Still, these p-values under 0.05 suggest to me that the ancestry of the Etruscan outlier was more complex than represented by each set of 3 populations tested, because not only do you generally want a p-value of at least 0.05, but also the higher the value, the better the fit. Still, other statistical tests tell us that she did carry an African component in her DNA, and the Moroccan Late Neolithic sample is, statistically, a better fit than Mata, so let's discuss the implications of that. One theory is that this Late Neolithic Moroccan genetic component could have come from contact with the Phoenicians who had settled in North Africa. The Phoenicians were an ancient Levantine group who were excellent seafarers, and they settled in many places around the Mediterranean, including North Africa, where they would have come into contact with the local populations. We know that the Etruscans were in contact with Phoenician colonies, so if the Phoenician theory is correct, then that African component in the Etruscan outlier was more likely derived from the late Neolithic Moroccans than from the ancestry seen in Mata, but this has yet to be confirmed. And as an additional treat, the study also published haplogroups. Mitochondrial haplogroups trace your direct maternal line, and Y-chromosomal haplogroups, which can only be tested in males, trace the direct paternal line. These are markers of ancient ancestry. All haplogroups have modern frequencies and a place of origin. Oftentimes their frequencies today align well with someone's ancestry, but sometimes they do not. Still, if we can get enough ancient samples tested and find out which haplogroups are common amongst different populations, we can use them as a proxy for migration when an incoming population carries different haplogroups from the population there before them. During the Mesolithic period, the only mitochondrial haplogroups represented are from the U lineage. 2 belong to the U5 sublineage and 1 to U8. These are fairly common amongst European hunter-gatherers and typical for the group. As for the Y-chromosomal haplogroups, all 3 hunter-gatherers were part of the I-M436 lineage, also known as I2A2, of which 2 belong to the I-M223 sublineage, also known as I2A2A. The I-M436 lineage has also been commonly found in the WHGs tested so far. As expected, we see a change in the mitochondrial and Y-chromosomal haplogroup profiles during the Neolithic. Mitochondrial haplogroup U doesn't disappear, but we also see the introduction of the K, H, N, and T lineages. Y-chromosomal lineages here include G2A, J, and R1b. It's interesting to see R1b in such an early sample because this haplogroup has typically been associated with the steppe expansions. And while DNA studies do show that male steppe herders carried R1b and contributed greatly to its high frequency in Western European men today, more and more ancient DNA studies are showing that R1b was present in Europe before the steppe expansion. Even some European hunter-gatherers carried R1b, even if at a lower frequency and not the specific lineage prevalent amongst the steppe herders. Still, it's becoming clear that a few prehistoric European men carried the R1b haplogroup without carrying any steppe-derived ancestry. We see more haplogroups getting introduced in later periods, but it's hard to assess whether this is a true introduction or not because of the small sample sizes. For example, the mitochondrial haplogroup HV doesn't pop up until the Imperial Period. However, we don't have nearly enough samples tested for preceding periods to know for sure that it was absent before that time. Looking at all the haplogroups listed here, overall, most lineages are Western Eurasian in origin and are found at high frequencies in Europe and West Asia. A couple stand out, however. Mitochondrial haplogroup D, which was reported in one individual from the Imperial Period, actually originated in Asia and is currently found at highest frequencies in that region, particularly in Northeast Asia, and is one of the 5 mitochondrial lineages found in Native American populations. The individual carrying this haplogroup had Eastern Mediterranean ancestry, where the D lineage is not as common. But as I explained, haplogroups are markers of ancient ancestry. You may not necessarily have any autosomal DNA associated with your mitochondrial sequence. The L mitochondrial lineage is also interesting here because it originated in Africa and is most prevalent there. However, L4 is a really rare branch, even in Africa, and is mostly found in the eastern part of the continent. It is interesting that it shows up in Rome during the late Antique period because that's when the general trend was a genetic shift towards Western and Central European populations. But the sample size is too small for us to definitively say that L4 was introduced during late antiquity. It could have been introduced at an earlier period, or maybe since it's so rare, it's unique to just a few individuals there. And that's it for this episode. Don't forget to give this video a like and subscribe for more cool content by your go-to informant on everything archaeology, Madam Archaeologist.